Hello guys, welcome back to another video tutorial from Shomu's Biology, and we are starting with the microbiology lecture series, and this is the first lecture of microbiology series. And in this lecture we are going to talk about the bacterial structure and function. So we'll be talking about all the different components of bacterial cell, including its cell wall as well as the extra cytoplasmic structures like fimbri, papili and all these things. So stay tuned and watch the lecture thoroughly. Now, first of all, let's start to talk about the characteristics of a living cell. Okay, so characteristics of a living cell can be like stated like all living things are made up of cells that share some common characteristics. So you can think about either a living thing can be composed of a single cell or it can be composed of multiple cells. So we call it single or multicellular organism. Now, the basic shape in this case will be spherical, cubical, cylindrical. An internal content of a cell composed of cytoplasm surrounded by a membrane, and for eukaryotic cells there are organelles, membrane bound structures which has a dedicated role to play inside a cell. In prokaryotes we don't have those organelles because in prokaryotes there is no particular fragmentation inside the cell that will do some specific job. So it's only just genetic material and cytoplasm and outside cell membrane. But the chromosome or DNA acts as a genetic material, and also inside ribosomes are there, as well as the metabolic capabilities obviously are involved. So based on all these cellular properties we can divide the cells into eukaryotes and prokaryotes. Eukaryotic cells are known as little updated versions which carry extra units, extra structures inside, extra organelles inside, while prokaryotic cells are primitive. They are earlier components. They are not updated enough. Okay, so here we want to talk about the prokaryotic cell. We'll be seeing all the prokaryotic cell types, their functions. Okay, so the eukaryotic cells example are animals, plants, fungi and protists that contain membrane bound organelle and that compartmentalize the cytoplasm or perform specific function, and they also contain double membrane bound nucleus, a specifically designed structure inside of which the genetic material of that organism, that is the DNA, or in much complex version chromosome, stays. While in prokaryotic cells like bacteria or archaea, no nucleus is found, no membrane bound organelle are found. You can clearly see the difference between the cell structure of prokaryote as well as eukaryote in this picture, where you can see in prokaryotes only genetic material floating in the cytosol covered by cell membrane. Sometimes they also carry cell wall covering outside, while in eukaryotes we have nucleus inside of which the genetic material is present. We have organelles like mitochondria, Golgi bodies, and also chloroplasts, lysosomes. So these structures, mitochondria, chloroplast, they are unique to eukaryotic cells only. Eukaryotic cells may or may not have cell wall. If eukaryotic cells have cell wall, those are generally the plant cells. Animal eukaryotic cells do not have cell wall. Now, if we start with the characteristics of life, which is pretty predominant, we already know that from our +2 level, but still, reproduction, genome composed of DNA packed into chromosomes, and that actually produce offspring, sexually or asexually. Reproduction is one of the prime characteristics of life, so as the growth and development. Metabolism, that is chemical and physical life processes inside the cell. Movement or motility, like a cell will move from its one location to the other. The motility can be like in terms of finding their food, or in terms of occupying a new niche, or for survival. Now the motility depends upon signaling, which can come internally or can originate externally. Cells support protection storage mechanisms like cell wall, vacuoles, granules and inclusions are there, and also transport of nutrients and wastes in and out of the cell. That's another very important important characteristics of life, because the cell needs to have a raw material for producing food. They need to have raw material for utilizing them to produce energy, and when they produce excess material, waste material, they need to be cleared out. So all these things are also present, and all of these properties are most important properties of life. Now, if we think about an overview of structural overview of a cell, if you look at this is the bacterial structure overview, you can see that this bacterial cell has a DNA inside, genetic material inside. It's floating in the cytosol. And if you look at cross-section view, it looks something like this, where surrounding of this nucleus there is cytosol, actually filled with mostly water, and there are other protein components present inside, and there are also different protein components, chemical components are there, and also small protein build structures are there to hold the cytoplasm structure very tightly. And this structures that is out there is formed by actin filaments. So actin are protein monomers which combine with one another to form this what is known as a cytoskeleton elements that hold the shape and structure of the cell by connecting this cytoplasmic components and other components of the cell with its membrane, and in turn membrane is protecting the whole cell from damage from outside. And maybe there are some cytosolic extensions. The extensions are known as fimbri, pili. As you can see, this is pili, these are fimbri, and flagella. All are the components which are extensions of the cytosolic components. For example, flagella is very big one, made up with flagellin protein in case of bacteria. On the other hand, this pili and fimbri, they are also made up with cytoskeleton elements, which has different role in terms of bacteria to be adhered to the surface of host cell to initiate the process of infection and causing the colonization. Okay, so they became a part of the outermost coating surrounding the cell membrane, which is also known as a slime layer, or which is like a glycocalyx layer, mostly made up with carbohydrate moieties attached with the membrane surface. External structures. There are two types of external structures that we generally found outside the cell. One are appendages and second is glycocalyx. As I mentioned, glycocalyx is nothing but a surface coating which is simply built with the help of some carbohydrate moieties that are fused with all the protein and lipid components of the cell membrane, while appendages are two different types. They generally help in either motility or attachment or as a channels. Motility like flagella, and axial filaments, which is also known as periplasmic flagella. Now both flagella and periplasmic flagella or axial filaments help the bacterial cells to propagate, to move from one place to the other, while others like fimbri and pili helps in the attachment of the cell with the surface of a host cell or host tissue. And this attachment or adherence is the very first step of the process of cellular interaction, bacterial cell interaction with the host, and that's the very first step of host pathogen interaction. So let's start talking about this flagella. Flagella has a very complex structure compared with this prokaryotic or primitive cell. Even though we call it primitive or prokaryotes, but flagella structure is so much complex. It's remarkable how the bacterial cell evolved to have such a complex structure. So it's known as a molecular turbine. So it is composed of three compartments: filament part, hook, and a basal body. The filament is a long thin helical structure composed of protein. As you can see, this is the filament region. Okay, this is composed of protein components known as flagellin protein. Now there is a hook. Hook has a curved sheath. This is the hook, which is a curved sheath like structure, which is kind of contractile in case of bacteriophage that we have talked about earlier. But in this case, the sheath also protects that filament because the filament is present inside of that sheath carving. The third thing is basal body. So basal body is the structure which is present embedded in the membrane of bacterial cell, and also cell and cell wall component structures. The embedding structure is known as the basal body. So the basal body contains the ring to the bottommost region and the rod in the middle. So with both the leaflet, both the side of the cell membrane, there is a ring. In the outermost leaflet, a ring is a smaller diameter. For the innermost leaflet, the ring is of higher diameter. But these two rings are connected with a rod structure inside. So that makes two different distinct component of the flagella, a stator and a rotor. So this is acting as a stator and the rotor is outside. So the stator remains inside, anchoring the flagella structure to the cell membrane of the bacteria, while the rotor unit continues to rotate, and as it continues to rotate, it generates a propelling movement that helps the bacteria to move forward towards a particular direction. Okay, it helps to move toward a specific direction, and this movement depends on a chemical signaling process, and that chemical signaling process is known as the chemotaxis. So chemotaxis is a process with which bacteria generally move towards a particular direction of a chemical component secretion. So that is the flagellar motility. So this flagella, with the help of this rotor unit which is present outside the flagella structure that we can see from outside, can rotate 360 degrees, and that is a beauty of this propelling effect, because it functions in motility of the cell through the environment. As you can see in this picture, that once this rotor unit continues to rotate, that allows the movement of the bacteria towards a direction. So in which direction? Obviously the flagella is present opposite to the head of the bacteria. In this case, this act as a head, this act as a tail of a bacterial cell. Now as the tail start rotating 360 degree, the bacterial cell will propel and will proceed towards the head direction, as well as a bacteria can have one flagella, it may have more than one flagella. Now this flagella can be present throughout of its cell, of its cell membrane. It can be anchored to different parts of their cell membrane. So that means the flagella can be rearranged depending upon their necessity. Okay, so in this picture you can see, in the bottommost picture here in the left hand side, we can see all the flagella is now arranged to one pole of the bacterial cell, so that they can be coiled and all the flagella can move to generate a much bigger propelling force, thus helping the bacteria to move forward faster in galloping fashion. While if the bacteria needs to float in one particular region, in one particular location in the aquatic environment, in that occasion the flagellal distribution took place throughout the cell, and as a result of which they simply propel themselves in their respective position, allowing the bacteria to hover and stay one particular place only. So this helps the bacteria to stay or to move at a particular direction. The flagellar arrangement, as we saw in this picture, are different, and this flagellar arrangement changes with time. So based on the arrangement of this flagella, we can divide the bacteria into different types. So like monotrichous, a single flagella at one end. Lophotrichous, small bunches emerging from the same site. Okay, so monotrichous, as you can see, this is the monotrichous. Lophotrichous is when all the flagella is moving out from one pole of the cell, which we saw here in the earlier picture as well, this one. Amphitrichous, flagella at both the ends. This is amphitrichous in this picture, this one, amphitrichous, because amphi means the flagella should come out from both the poles of the bacterial cell. And peritrichous, the flagella dispersed over the surface of the cells, throughout the surface of the cells, that is peritrichous. So this one was peritrichous, this one was monotrichous. This will be lophotrichous because many flagella all together, and this is monotrichous. Okay, so mono and lopho both is a situation where the flagella is present at one pole of the bacterial cell only, but monotrichous is when there is only one flagella unit, and lophotrichous is where small bunches of flagella together. Flagellar responses. Now the flagellar response can help the bacteria to move towards the direction of a chemical secretion via the chemotaxis mechanism. Now that will allow the bacteria to move. Now the bacterial movement can be straight, but generally the bacterial movement always is in zigzag line, which is unpredictable, as you can see in this picture below. So you can see that either a bacteria can hover at one particular position when the flagella is equally distributed, okay, which is known as peritrichous situation. But in a situation where the bacteria to progress forward, they need to have the flagellar arrangement to one pole. So the chemical stimuli is required, which is known as a chemotaxis process, that can be of either positive and negative stimuli. Now the light stimuli is known as phototaxis, and the chemical stimuli is known as the chemotaxis. Now the signal sets flagella into motion, either clockwise or counterclockwise motion. If the motion is counterclockwise, that results in smooth linear direction. If the motion is clockwise, that results in tumbling motion. So either, as I said, a straight motion or a tumbling motion, zigzag motion like this. So it's not even zigzag, because zigzag means changing direction alternatively. That's not the case. Tumbling means you cannot predict the movement here. So if it's counterclockwise, then in that case, so you can imagine this is counterclockwise. Counterclockwise means in this case it is a smooth unidirectional linear motion, and clockwise means tumbling motion. Okay, so remember that. And there are sometimes periplasmic flagella to be present, because we know flagella is an extension of the cytoplasm, and that is actually originated from inside of the cell. Although it's originated from inside, but the flagella's most of the component is present outside of the cell. So this is kind of connected to the cell membrane and ultimately connected to the cell cytosol from inside. But there are flagella that is present near to be connected. You can see that internally. So we call them internal flagella, enclosed in the space between outer sheath and the cell wall peptidoglycan. So we know that two different types of bacteria are generally found. One with a very thick peptidoglycan layer, another one with a very thin peptidoglycan layer. But no matter what situation it is, that outer sheath of this flagella can be present in trapped with this peptidoglycan layer. So we call them internal flagella. It produces cellular motility by contracting and imparting twisting or flexing motion, and that can help in like generating a corkscrew like twisting motion, and that helps the movement and spiral movement of the organism where the flagella is present. And there are examples found in few occasions the flagella to be having like this. We call it periplasmic flagella. You can see in this picture, this is a periplasmic flagella that we can clearly see. Okay, you can see this is the cell membrane, and this is the outer sheath, and this is the periplasmic flagella that is kind of surrounding the whole cell. If you look at a cross section, it looks something like this. Otherwise, it's something like there is a big rod and a rope is tied across that rod in such a way that in every single time if you do a cross section, you are going to see some portions between the cell membrane as well as the outer sheath layer. Now, apart from flagella, the other cytosolic extensions are papili and fimbri. We know flagella helps the cell to move. It helps in the motility, and it's mostly composed of the protein flagellin. Fimbri is also fine proteinaceous hair-like bristles emerging from the cell surface, which you can clearly see in this fluorescent diagram of microscopic image. You can see that these are the bacteria, and these are this fimbri that is going out and sticking out from surrounding this bacterial cell all the time. Okay, it functions in adhesion to other cells and surfaces. So this fimbri only is helpful for the bacteria to be adhered to the surface of the host cells, and this adherence help this fimbri to stick to the host cell and start colonization process quite fast and quite well. That's the idea about fimbri. Similarly, there is another structure called pili. This is a rigid tubular structure made out of another protein known as pilin, found only in gram-negative bacteria. Function to join bacterial cells for particular DNA transfer called conjugation. Now think about it. All these structures are extracytoplasmic structure. So the structure is an protrusion out of the cell, but they have different roles to play. Like flagella has a role for a propelling movement of a bacterial cell, while fimbri's job is to attach and adherence of the bacterial cell surface with any inanimate structure where they need to colonize and form biofilm like structures, or to adhere to the surface of the host cell so that they can initiate the colonization. But on the other hand, pili, which is only found in gram-negative bacteria, has a different role, and a role of genetic component transfer between the cells of their same generation, which is also known as horizontal gene transfer, HGT. This horizontal gene transfer is a process with which the bacteria can share genetic component with themselves, with the member of their same generation, and that is really helpful in terms of sharing a very important genetic component, in terms of sharing the genetic material between themselves, and that helps them to initiate and get important properties like antibiotic resistance properties. Talking about the layers on the surface of the bacterial cell, another such layer is glycocalyx. Glycocalyx layer is just like a slime layer on the surface of the bacterial cell. It's not like any protein component protruding out. It's a coating of molecule external to the cell wall made up of only sugars and or some proteins, but mostly this component is made up with sugar component. And there are two types of glycocalyx that are found, either a slime layer or a capsule layer. Both of them are filled with these sugar moieties attached with proteins, attached with the fatty acids. So slime layer is loosely organized structure and attached, while capsule is highly organized and tightly attached structure, but the structural components are kind of equal between the slime layer and the capsule. Now the component of slime layer and capsule help this bacteria to escape the host defenses, because the host produces antibody to bind with specific antigenic determinant which are present on the surface of the bacterial cell. Now this glycocalyx, like slime layer or capsule, is covering all the antigenic determinants in the surface. That's going to prevent the antibody to properly bind, and thus it prevents the killing of this bacterial cell by the host immune defenses. On the other hand, this glycocalyx layer also help this bacteria to properly interact and adhere to the again surface of certain inanimate component as well as some other components in the host cell surface as well. Now, as I mentioned, two functions, but you can say protect the cell from dehydration and nutrient loss. This is another important feature. Inhibit killing by white blood cells by phagocytosis. So it's contributing to pathogenicity, right? And the attachment. So this glycocalyx layer produces so many carbohydrate moieties on the surface, which is very good in terms of creating a long lasting, very tight and very strong interactions between the inanimate component, between the environmental components of the bacteria, thus forming biofilms. And we know biofilms are so much complex in terms of its adhesiveness that we cannot even scrap out the bacterial cells out of the biofilm quite easily. That's why you have the biofilms in your tooth. We simply cannot scrap it out that easily. So what is this biofilm? Biofilm on a catheter. You think about this is a biofilm on a catheter, but in other examples we have biofilm in our tooth, we have biofilms in any place where there is a constant movement of liquid containing nutrients. So you can see this is the catheter surface, and we can clearly see the catheter surface. This is the catheter surface, the black colored, and this is the biofilm made up with bacterial composition and fungal cells as well. So some of them are fungal cells, and this is staphylococcus cells. So staphylococcus and fungi both remain together in a positive reinforced positive interactions between them on the surface of that catheter surface. So this is just an example of how these bacterial cells like staphylococci properly adhere to the surface of catheter surface. And similarly, the same thing for other bacteria that generally forms biofilm in our tooth. And how they do this? Because they have those carbohydrate moieties, and with the help of this carbohydrate structure, it's like a glue with which they can easily adhere and attach to the surface of our tooth. The cell envelope. Now, till this point, we've been talking about all those extracellular components, right? We have been talking about those glycocalyx layer, the flagella, fimbri, pili, all this. Now, there can be also cell envelope. Okay, this is another extracellular component. External covering outside the cytoplasm, composed of two basic layers though, cell wall and cell membrane. And we know both of them are most important in terms of providing safety and shape and structure to a cell. So generally not all the microbes have a cell wall, but all the microbes definitely have cell membrane, because they need to have cell membrane to maintain the cell as a separate system, as a closed system. They maintain cell integrity. And there are two different groups of bacteria demonstrated by the Gram stain, which actually kinds of help us to find out the presence of thick or thin cell wall. Now those with very thick cell wall is known as Gram positive bacteria, because it's composed of primarily of peptidoglycan as well as cell membrane, while the other one with very thin cell wall known as Gram negative bacteria, they have a very thin peptidoglycan cell wall layer, although they have a third and an extra layer of cell membrane outside. We call them Gram negative bacteria. So this is the onset of understanding Gram positive and Gram negative bacteria, although the nomenclature is given based on how they respond in terms of Gram staining, which is in turn is named after the name of the scientist who actually worked with the staining process and principle. So Gram positive, they have a thick peptidoglycan cell wall. They don't have any third cell membrane outside. Actually not third, it's a second layer of cell membrane outside. While Gram negative bacteria, they have an extra layer of cell membrane outermost region, but they don't have very thick peptidoglycan layer. They have a very thin peptidoglycan layer. You know, in bacterial cell structure, we don't focus mostly on cell membrane and other cellular components like cytosol, because this is very similar with that of the eukaryotic cell. The major difference was in the cell wall, and the difference is with the outer cellular component, like extracellular components like glycocalyx layer, the capsules, the slime layers. Okay, so we have talked about that. So the cell membrane is also made up with phospholipid bilayer with a mosaic pattern of protein embedding like a mosaic pattern. And this cell membrane functions in providing site for energy reactions, nutrient processing and synthesis. It is also acting as a passage of nutrients into the cell and discharging wastes out of the cell. And the cell membrane is selectively permeable, so it will allow only certain molecules to move in and out. Now if you think about the inside of the bacterial cell, we only need to talk about two things. One is the cell cytoplasm, and the second one is the genetic component of the cell, that is bacterial chromosome. You know, the cell cytoplasm is a dense solution of sugars, amino acids and salts that are present, and it is composed of mostly water, 70 to 80% of the water. It serves as a solvent for materials used in the cell functions like the proteins, like amino acids, sugars and salts. So this act as a medium, as a solvent for all those components. Now the last thing, nucleoids like chromosomes, plasmids, and also ultimately the bacterial chromosome. The bacterial chromosome can be single, circular, double-stranded DNA molecule that contain all the genetic information required for the bacterial cells to survive. That is the bacterial chromosome. Apart from bacterial chromosome, bacteria may have plasmids, which are very small, circular, double-stranded DNA which is capable of self-replication, means autonomously replicating themselves. So not essential for bacterial growth and metabolism, but they can provide the bacteria extra benefits or extra properties like antibiotic resistance feature, like utilizing certain or like helping to produce certain enzymes to utilize a nutrient the bacteria generally is unable to utilize. So those are the roles of plasmids, and bacteria generally utilize the fusion for its division purpose as well as the process of conjugation between themselves. So in all the occasions, plasmids can also be transferred from one bacteria to the other, and that acts as a gateway of conveying the message and sharing important information like the antibiotic resistance feature from one bacteria to the other bacteria. And these plasmids are blessing for us as a molecular biologists during genetic engineering to readily manipulate and transfer from cell to cell. Other than that, there are bacterial ribosome, which are little different in structure and its function compared to the eukaryotic ribosome. While eukaryotic ribosome is known as 80S ribosome, the prokaryotic ribosome is known as 70S. Two components known as 130S, the large subunit, and the other one is 150S, the large subunit, another one is 30S, the smaller subunit. Now, this ribosome involved in the protein synthesis, and bacteria continue its protein synthesis inside the cytoplasm because they have the ribosomes inside the cytoplasm always, and they made 60% of ribosomal RNA and made up with 40% of sole proteins. Okay, prokaryotic are different in terms of ribosomal structure with that of the eukaryotes, and the size of the ribosome is also little different. And this ribosome is found in every single cell of prokaryotes because it is so essential that it's required to produce proteins that helps the bacteria to survive. The other bacterial internal structures like inclusions and granules. Like intracellular storage bodies, they can be vary in size, number, and content. Like bacterial cell can use them when environmental resources are depleted. So this is also there, but it's also very, very rare. Not like the plants, because in case of plants, the vacuole is always present, and the vacuole's volume and size is huge always because it stores a lot of secondary metabolites and other metabolic components inside the vacuoles to use them in future. While in bacteria, it's not the case like vacuoles, but still they have vacuole-like intracellular storage bodies, which are varying in size, number. But they can store other components inside that can be used by that same bacteria while the environmental conditions are turning harsh. The other internal structure of bacteria which is really important is cytoskeleton, which I mentioned earlier as well. Cytoskeleton like actin filaments, which holds the cell membrane and connects it with the rest of the cellular components inside, and it actually literally provides a skeleton to the cell so that the cell has its own shape. Otherwise the cell should collapse and should not have any particular shape. So many bacteria possesses an internal network of protein polymers. They are closely associated with one another. Particularly in case of this bacterial cell, there are microfilaments or actin filaments. Okay friends, so that's all about the bacterial structure and function. See you in the next lecture of microbiology. So if you like this video, please hit the like button, share this video with your friends, subscribe to my channel to get more and more videos like that.